So, greetings everyone from whichever part of the world you are, or if you happen to be watching this recording later. Thank you all for coming today for our first PHIM Club event of the spring 2026 semester. So, today we will be hosting a very special guest speaker session by an ASU professor, Dr. Judith Klein-Seetharaman, who is part of ASU's School of Molecular Sciences and College of Health Solutions. She obtained her PhD with late Nobel laureate Hargobind Khorana at the Massachusetts Institute of Technology on conformational changes and folding in the G protein coupled receptor rhodopsin. And she also holds dual undergraduate degrees in biology and chemistry from the University of Köln. I hope I'm pronouncing that correctly, Germany.
Köln. It has umlaut, two dots.
Right. So, Professor Klein's research combines computational and experimental studies of protein structure, dynamics, and function with particular emphasis on membrane proteins. She has published over 150 papers and has received a number of awards, including the FP7 People Funded Marie Curie International Incoming Fellowship, the Sofia Kovalevskaya Award of the Humboldt Foundation, a Bill and Melinda Gates Foundation Grand Challenges Award, National Science Foundation Career Award, and the Margaret Oakley Dayhoff Award of the Biophysical Society for her remarkable work in computational biology, embracing the full spectrum of experimental biophysics. And we're excited to host her today. So, our agenda will be a 45-minute talk approximately, and after that, we'll open it up for questions. So, if anyone in the audience has questions, feel free to ask them at the end, and you can unmute or ask them in the chat. And to you, Professor Klein.
All right. Thank you very much for the kind introduction and for inviting me to speak here today. Um, if you do have questions in between, you want me to fast forward or expand on anything, please do let me know. I'm not exactly sure whether my talk will be exactly 45 minutes, so I might start cutting down a little bit in the end. So Vina, please let me know when it's starting to become too long. Um, so um, so Vina has already said a lot about my career, but she asked me that I should speak about my career. So I'm going to give you a little bit more details on who am I. And so let's see, why is this not advancing? Hold on. Okay. Let me move this somewhere. Okay, so you will see that I'm really interested in kind of a data-driven discovery in biology because of my interest in combining computation and experimental approaches. And so a lot of my work is method development driven, and we try to use those methods to address interesting questions that we then plan to validate in the lab. And so I'm going to tell you first a little bit how we got there, and then I'll give you an example of this approach, and then I'll tell you what we have been working on recently. So, like Vina said, I did my PhD with Hargobind Khorana, and that really determined, I guess, a lot of my interests and future research. So I'm going to tell you a lot about that time, and before I move on to some more recent things. So here's an overview of my scientific career timeline. So I was born in 1971 in Germany. And yes, Cologne is Köln. So I was educated. I went to school in Germany, and I also went to university in Germany. So I got my undergraduate degree. But already when I was an undergraduate student, I um, I I wanted to expand the discipline that I was studying. So I wanted to study biology because at that time there was a lot of environmental disasters, and I was really upset about, for example, oil spills that killed a lot of birds and things like that. And I felt like it was the chemistry's fault that there was so much damage done to the environment. And so I decided that I should really know more about chemistry as well. And the chemistry curriculum for biologists in Germany at the time was very, very minimal. So there was basically one class, one semester that combined all of inorganic, organic, and physical chemistry. And so I decided that I just enroll as a chemist too. And so what I did then is do two full undergraduate degree courses in parallel, because at that time we didn't have all these interdisciplinary subjects that we have today. So at that time you had to choose, you either want to be a chemist or you want to be a biologist, and there was no interdisciplinary options. And so this is what I did. And so then I finished my undergraduate studies with a research project that was on photosynthesis. And in my hometown in Germany, there was no biochemist researchers at all. So I actually had to move somewhere else to be able to do research in that area. And so I moved to Imperial College in London, where I did a project on photosynthesis. And that's where I first learned about proteins and conformational changes. And at that time, conformational changes was used as a kind of magic word that was used to explain like a black box when a protein had different functions. And so one day when I was sitting in the library reading about proteins and conformational changes, I found this paper on bacteriorhodopsin. And this was a paper in which the structure was described with great detail on where exactly a proton is moving from one amino acid to the other. And I was really amazed that you could have such great details. And so I wrote to the senior author of that paper, which was Hargobind Khurana, and asked him that I would like to do my PhD with him. And so he then invited me to meet him when he was visiting the UK, and eventually I applied and was admitted to MIT, where I then did my PhD with him, and I'll tell you more about that time. And so I'll jump now to telling you a little bit about this, so you will see the rest of my career later. So here is an introduction to Hargobind Khurana. So when I wrote to him, I was very ignorant and naive. I had no idea that he was actually a Nobel laureate, and actually only found this out after I had joined his lab. But he has made absolutely seminal discoveries in biology as a chemist. So he's an organic chemist by training, who through chemical synthesis has been able to decipher the genetic code. So when you learn in biochemistry class that triplets encode amino acids, it's because of Hargobind Khurana's work. And he shared the Nobel Prize with two other scientists as well. And so this key insight into the biology of genetics, really, he translated into methodology. And I find this extremely important, that it's not only the curiosity about biological questions that are important, but also how you can make use of this information to get more information about scientific systems. And so he exploited this information by actually carrying out the first total synthesis of a synthetic gene. And so this was originally a tRNA, but that's not directly relevant to my thesis. Instead, he also did this then for bovine rhodopsin, and bovine rhodopsin is the protein that I did my PhD on. So this is this protein here on the right-hand side, and you'll hear a little bit more about it. So this was a key insight translated into a novel methodology that opened up the universe of manipulating proteins, because up until then, you painstakingly had to purify this from original systems, and you wouldn't be able to manipulate it in any way. Whereas with a synthetic gene, you can replace the amino acids, introduce mutations, and then study the effect of the mutations on the structure and function of that protein. And so this is a picture from the time when I was in the Corona lab. So that's like around 25 years ago, I guess. So these are all our lab members. And I wrote a recent obituary for Gobind, because he would have been 100 years old in 2023. So if you're interested, I have lots of stories in that article, which was published in Biophysical Review. So that's this picture here. Yeah, so I just want to tell you one of those stories because it is quite amusing. I mean, not everything doing a PhD with Gobind Khorana was amusing. He was pretty tough. In fact, many people called him a slave driver, and he was pretty tough. So I'll give you one amusing example. So every Saturday morning, we would find this box of donuts in our group seminar room. And Gobind would always come in on Saturdays because he didn't want to tell us specifically that we should be coming in on Saturdays. He carefully had orchestrated every week to be such that there was a seminar on Monday morning, another seminar on Wednesday at noon, and a third seminar on Friday afternoon that finished around 5 to 6 p.m. So by definition, you could never travel because you had to be there for those 3 seminars. But he did not require us to come in on Saturdays. And to reward the people that did come in on Saturdays, we received donuts. So after some time, I realized that actually the donuts had a secondary motive. So in fact, the donuts in Gobind's brain were translated by his lab members. And so we realized that because there is a dozen of 12 different donuts, the preference of each donut by a person indicates who was there on Saturdays. And when we figured this trick out, people in the lab would call those that were present, for example, me. I would receive a phone call and say like, Judith, can you please eat my Boston cream? And so we were very much motivated to work very hard and produce lots of results. And this is just one of the tricks that he used to accomplish that. And so it was a very productive time. So out of my PhD, I published 16 publications. But this is one of the lessons that I've learned during my PhD, is actually that you sometimes need very powerful friends to help you in your career development. So so as a PhD student, you have to form a PhD committee, so the committee that keeps track of your progress and eventually helps in deciding when you can graduate. And originally, Gobind wanted to have a representation of his close friends and colleagues on my committee. And I said, like, I would rather like to choose my own committee members. And so I chose my own committee members, and it turned out that that was very, very useful because I had wanted to graduate, and Gobind always said like, no, you still have so much work to do. You need to work more. And so eventually at my committee meeting, I made a list with all the things that I had done and the list of the things that I felt I can still finish before I really want to graduate. And so the latest result that I had received at that time was the first ever isotope labeled NMR spectrum of rhodopsin, and rhodopsin as a G protein coupled receptor, the very first NMR spectrum of a G protein coupled receptor ever. And so this is the spectrum, the 2-dimensional so-called HSQC spectrum of rhodopsin that was labeled with N15 lysine. And so you can see here on the x-axis the proton chemical shift and the nitrogen chemical shift on the y-axis, which indicates whenever there is a spot, it represents an NH constellation. And this is the spectrum that we got. And so at this committee meeting, Goben said, well, Judith, I don't think you can graduate. You need to assign these peaks. How do we know what lysine is each of these peaks? And so one of my powerful committee members stood up and said, no, Gobind, she doesn't. And so it was only thanks to her that that I was allowed to graduate. The committee agreed that I did not have to assign these peaks and I could write up my thesis. So just so you know, I graduated in 2001, and it was actually only more than 6 years later that future students of mine and my collaborators' own labs managed to finally assign these peaks from my thesis. So if it had been if I had to follow Gobin's demands, I would have still been a PhD student for 6 more years, which would have made it I don't know, 12-year PhD or something like that. So so that's just for curiosity. So this is a picture from that 2007 lab that shows that it's assignment, and this was actually quite a complex and very labor intensive project that led to this. So the topic of my thesis, as you probably have gathered by now, was rhodopsin, and so I worked on dynamics of rhodopsin. In particular, I was interested in conformational changes in result of activation. So in the case of rhodopsin, it is light activation, but in case of other G protein coupled receptors, it can be activation by ligands. And so ligand binding is really important for protein function. And so this has really been a theme throughout my career, trying to understand what does ligand binding do to protein structure and how does that relate to their function. And in all aspects, the motions of the protein are really, really important. And so I've developed a lot of methodology to be able to study these kind of things, because these are very challenging systems, because this is a membrane protein. Like Mina said, my main interest in the protein fields. I like all proteins, so I'm not very discriminative, but my favorite proteins are membrane proteins, but they're actually experimentally very challenging to study. So there's always a lot of method development that goes into being able to study these kind of things. And so in the year 2000, this was actually, 2001, this was actually also the year of the first publication of the first human genome sequence. And so this was a paper published in Science, and it was hailed as the Book of Life. And so this was really an important milestone in my career, because this analogy opened up the interest of computer scientists who had worked on human languages before and thought like, oh, maybe the techniques developed for human languages can be applied to biology. And so at the time, I had the two-body problem. And if anybody is interested in more details about that, I'm happy to speak about it later. So my husband had already moved to Pittsburgh while I was still a PhD student. And I was looking for a reason to also move to Pittsburgh scientifically. And so I then moved to the School of Computer Science at Carnegie Mellon University, because I found out that Carnegie Mellon was the #1 in the country in computer science. And so I really wanted to do something with computer science. And so, this analogy between biology and language really shaped my career, because it allowed me as a biologist and chemist who had never done anything with computer science before to start collaborating with computer scientists who had never done anything in biology or chemistry before. So we actually had a lot of fun cross-educating each other. I learned programming and they learned about the genetic code. And so we ended up developing this approach very extensively and received a large-scale center grant from the National Science Foundation called Biological Language Modeling. And ever since then, basically, I combine computational and experimental techniques. And I love collaborating with people from other domains. And so, so along the publication of the human genome is really it marks sort of a milestone in the data revolution in biology, which is moving, which started to move already at that time into generating large amounts of data, especially sequence data. So the human genome sequence was maybe the 50th or so genome, besides quite a lot of microbial genomes that were first sequenced. And since then, it's been exploding, as I'm sure you're all aware. But alongside has been the whole pipeline from DNA to RNA to protein to modified proteins and to protein-protein interactions and the molecules that are manipulated by these proteins. And so we moved from individual molecules to what we call systems biology, so the bird's eye view of many molecules at once. And so the processes that were originally described as transcription translation, they became then from transcription to transcriptomics, and individual proteins became proteomics, and individual metabolites became metabolomics. And so, so this is really what we are trying to do, is actually try to understand how a system works with all these individual components. And so I'm going to just give you a recent example how this approach turns out to be. So instead of having sitting there like traditionally, you ask yourself a question like, what am I interested in? And I'm gonna develop a hypothesis what this protein might be doing or something like that, and then you will design some experiments to do that. So this approach is we are collecting this incredible amount of data, and what do we do with it? And so, so basically, we use that data to develop methodology that generates biological hypotheses. And because of the very large amount of data, these hypotheses are now completely novel and are typically things that a human brain by itself cannot come up with. And so, so I'm going to give you some examples for this. And I'm going to give you a recent example because, as I mentioned, you know, in 2000, it was the sequencing that started to explode. And in very recent years, it's actually the protein structures that have exploded. And in particular, the ability to connect them. So because of this incredible amount of data, we are now able to predict protein structures in many cases with very high accuracy from sequence information alone, which reduces vastly the amount of time, effort, and money that you need to spend to actually develop the 3D structures of proteins that you might be interested in. And this is really what we are interested in analyzing. And I'm going to explain to you so just like how transcriptome becomes transcriptomics, I then use the word targetome because, as I mentioned, in the case of GPCRs, the ligands that proteins bind to really determine their function. And so that's what we are really interested in, is when you have a ligand, and that ligand can be a small molecule or it can be another protein, you want to know where does it bind. And now with this explosion of structural data, we can start asking novel questions such as, what are all the possible proteins that a ligand might be binding to? And that's the universe of all targets, and therefore I call this the target. And so, so basically we are extracting information, and it's critical that this large amount of data is publicly available. And so the main go-to location for us is the protein data bank. Anybody that determines the structure deposits their structure files in the protein data bank, and it is free to use in research. And so we extracted all the proteins from human. So because we first asked the question, what is the human targetome? So what are all the human proteins that molecules might be binding to? And it turns out that out of the 22,000 proteins in the human genome, more than 7,600 have at least one structure deposited in the protein data bank. And so this is the data set that we have been using. And then the next question is, well, what ligands are we talking about? So many of you have probably heard about drug discovery and the importance of drugs for medicinal chemistry, where the traditional pipeline is that you choose one protein as your target, you validate that that's an important disease target, and then you screen millions of ligands against that one target in order to find the best possible ligand. And that's then your drug that goes into the drug discovery pipeline. And so I'm asking the reverse question. I'm asking, well, what about all the molecules that we are encountering on a daily basis? And these can be metabolites. So these can be molecules that the body makes and transforms itself. It can be what it takes in as food. So for example, glucose, so sugar, is an important food ingredient. Yet we know that we need sugar, but we know that too much sugar is actually bad for us. So you see already the duality in the function of these molecules. It can be good for you, but it can also be bad for you. And so those are all really interesting questions that I want to use this data-driven approach to actually try and gain some new insights into what do all these molecules do. And so like how glucose can be bad for you, there's of course many other molecules that we know are bad for you, such as many pollutants. And so here's one example called benzoapyrene. This is a very ubiquitous pollutant that you can find in water, soil, air, and it causes cancer. So it's definitely a really bad molecule. And so we can start asking the question, like, how do these targets differ from each other? Then, of course, we have vitamins that you can eat through food sources or through supplements. And you have many more molecules that are in natural sources. So this is an example from lemon juice. But as Veena and many of our students know, there's a universe of many molecules that have biological effects and medicinal properties from various natural sources, especially plant sources. And much of that remains to be discovered. And even when molecules have been structurally identified, like this one, oftentimes it's not known what the targets are. And so those are all questions that we can start addressing. And then last but not least, there is the traditional drugs. So an example is metformin. This is an anti-diabetic drug, very successful drug. But when we look at which is the target, it's actually not known. There have been a number of targets that have been proposed, and it turns out that this is something called a dirty drug that binds to many different targets. And so having this resource of structures available allows us to start asking this question, well, what are all the proteins that are impacted by this and other molecules? And then we can also start asking the question how they impact each other. Maybe if a binding pocket is already occupied by large concentrations of a metabolite, it won't be accessible for another molecule anymore. And so those are all what I find interesting questions that one can address. And how do we do this? So we use a computational approach called molecular docking. The method, specific method we have been using a lot is AutoDock-Veena, which predicts the binding. And this is a picture from Veena, not that Veena, but this Veena, your host Veena. So this is on the x-axis are the protein data bank identifiers for those 7,600 proteins for which structures are known. And then on the y-axis is the predicted affinity. So the strength of the binding of ligands to those proteins. And what's shown in blue are all the different ligands that we've already subjected to this molecular docking approach. And so the lines here look blue, dark blue, because they're actually very faint light blue, but there's more than 1,000 ligands that we have been docking. And so you see where the majority of the ligands lie with respect to their relative affinities. And this is sorted by one particular ligand. So you can ask the question, what's the best target for that ligand? And what's the second best and the third best? And you see that when you expand this, that there's typically quite a few targets and not just a single target that has very high affinity to a particular ligand. And so we've been doing a lot of comparative analysis for these types of ligands to try and see how they impact each other. And so here I want to give you an example for serendipity in my career and in science, because when I was analyzing this data, like I said, I'm really excited about data-driven approaches. So basically, looking at this figure, there is no hypothesis that I had here, okay? I just asked the question, where do all these molecules bind? With no implication, with no hypothesis as to where they would be binding. Just completely open to anything. And so this is the result. And so then when you get a result like this, you want to analyze it, right? So I zoom in to this region here with the best possible affinity. And lo and behold, what do I find? This protein called 6FU. This is the identifier, okay? 6FUF. Doesn't mean anything, right? It's just the identifier. But I look it up and it's bovine rhodopsin. How can it be that bovine rhodopsin, the topic of my PhD thesis, is even in our data set? Because our data set is human proteins. It shouldn't be there. Nobody has ever crystallized human rhodopsin. So how can it even be that rhodopsin is in there? Bovine rhodopsin is even in there. Turns out that this particular protein is a fusion protein of bovine rhodopsin with human G0. Because rhodopsin is a G protein coupled receptor, it binds and activates G proteins. And so somebody studied this complex. And because it was human, it appeared in our data set. And it was amazingly the top predicted target for benzoylpyrene, that pollutant. And here I am 25 years after my PhD. Okay, it's binding to rhodopsin. Okay, let's study it more. Very, very, very interesting. And so not only does the pollutant bind, but it turns out that the lemon juice molecules, some of them also have very high affinity. But if you look at the lemon juice molecule here, you see that there's the red ones are lots of oxygens, right? So this is a molecule that's extremely hydrophilic. It loves to be in water, whereas look at benzoylpyrene, it's very, very hydrophobic. So I was like, how can 6FUF be a target for such molecules with such different physicochemical properties? And it turns out that the lemon juice molecules don't bind in the same pocket where benzoylpyrene binds. Instead, it binds over here. When I saw this pocket, I was like, oh my God, I know this pocket. And here's this pocket. Many years ago, I studied this really interesting phenomenon of chlorophyll binding. So somebody had, and there's a really, you guys should watch it. There's a really amazing movie, so a documentary series about animal life on the planet with David Attenborough narrating. And there's this amazing video of this deep sea ocean fish called Malacosteus niger, this very scary looking fish, which has this light under the ice. So in the depth of the ocean, it's pitch black, no light at all from the sun. So the light that is there is generated by bioluminescence. Most of the bioluminescence in the ocean is blue or green, but there are some exceptions to this. This fish is one of them. It makes red light. And in fact, it has its own flashlight under the ice that shines red light. And because all the other organisms can only see blue and green light, they have no idea that Malacosteus is illuminating it. There's this famous movie where you see this little tiny copepod swimming in front of the thing and eaten by Malacosteus niger. So that was a really fascinating story for me. And I was like, wow, so how does this work? I mean, how do they detect their own red light? And so there was already some research into that, and people could not find, for the life of them, a gene that would encode a rhodopsin with the right absorption maximum. So the hypothesis was proposed that this is all the algae that are dying in the ocean. There's all this chlorophyll molecules around, and they're the ones that are conferring this red light sensitivity to this fish. So okay, let's take that. And so we studied this molecule. Actually, here you can see it, this one here. It's called chlorine. So chlorine was the one that was identified as the best in enhancing sensitivity to red light. And so we did a lot of experiments, starting with molecular docking, that showed that it binds in the cytoplasmic domain. And that's exactly that pocket in the cytoplasmic domain here. Sorry, the protein is upside down here. That's why it's upside down. But this pocket is actually this pocket here. So that was really amazing to me, because we actually published several papers about that pocket, which was the first ever allosteric ligand binding pocket identified for, oh, sorry, that was my dog, in rhodopsin. So this was really, really exciting results that we could not have anticipated, and that came out of just looking at the data innocently without any bias, and just checking what can you find. All right, so I know I only have a few minutes left, so I'll just briefly tell you about our current focus on aging. So I've been moving from this very fundamental interest in protein structure, dynamics, and functions into doing things that may have some benefit for people. And so I was alerted to the so-called X Prize Health Span competition now almost exactly 2 years ago. And so this is a global competition with the goal of turning back the clock, the biological clock of aging, by 20 years in a time span of 1 year. That's the very lofty goal, and it's extremely popular. This is the largest competition that this organization, this nonprofit organization called X Prize, has run. It's worth $100 million. So if we're actually able to do this, and if we're able to do it better than everybody else that's participating, we will get $100 million. But unfortunately, it's a very tough competition. There's 765 teams globally that are competing for the same prize. So this is a very, very, very competitive project. And so why am I doing this? So here you can see I'm not that young anymore. So I was like, I guess it's maybe time to do something about it. And the other nice thing, this competition focuses on an integrative approach. So the idea is not, oh, we need to cure diabetes when we find it, or we need to cure Alzheimer when we find it. No, it's about aging affects many different parts of the body simultaneously and increases the risk of diseases in these multiple systems, yet they have shared mechanisms of disease that could be tackled maybe by one or a common set of interventions. And that's really nice, because as you probably have realized by now, I'm a very unfocused person. I cannot be pinned down to work on a single disease. I never wanted to do that. And here I have the perfect excuse to be absolutely unfocused and work on all the different diseases, because many of them are age dependent. And so, so that's what we are doing here. So me being 54, clearly I'm old. And so the idea is turn back the clock as far as you can. So the first question I asked, like, well, I have a daughter that's 13 years old. Can I be like her? You can see she's extremely flexible, and I am extremely unflexible. So those are all things like, yeah, there's lots of really functionally desirable properties that you would like to have as a younger version of yourself. And so as a molecular scientist, what does that translate to? So, so we actually in the lab have been working on identifying these molecules that the human body is encountering. And one of the approaches to do that is called metabolomics, right? So the science of studying as many molecules from a particular blood sample, for example, or other body sample as possible. And so this is in collaboration with my former postdoc, Panis Jaspi, who has founded a company called Therium that actually commercialized this. He made a kit out of an assay that is metabolomics based. And so I gave my blood sample to him and my daughter's blood sample to him. And here's what he gave back to me. So metabolomics is a very data-intensive approach, of course. In this case, it has 126 metabolites, and so it's definitely information overload. So he has developed a digital twin that takes all the papers that have already been published on metabolomics and integrates them to associate these metabolites with specific disease or health related areas. And so he came up with this score where red is poor. So anything 4 or less out of a scale of 10 is bad, is red. Yellow is kind of bad, but not that bad. And then green is good. And so now I'm asking you the question, do you think that this is me or this is me? Who is Lamia and who is Judith? Does anybody want to guess? I guess Vina told you are not allowed to say anything. So, okay, fine. You can put it into the chat and we do a count later how many people think this is me and this is Lamia. And so, in any case, can I become like Lamia? And so this is really the background of this approach is, can we turn back the clock of aging? So it's like time travel. Can I be a younger me? And so there's actually evidence that you can do that. So this is an example where people are no longer caring about what your actual age is based on when you were born, but based on what you might be caring about, like your fitness. And so people are calling it the fitness age. And there's all kinds of age clocks, like metabolomic age and whatever. So there's many different types. And fitness age is one of the ones that many people care about. And the parameter that's measured here is VO2 max. And so I'm running out of time, so I just want to tell you briefly that here's a graph of VO2 max over age. So you see how VO2 max, a fitness parameter, decreases with age. And so if you can improve your VO2 max, increase it, then basically that means you'll have a younger biological age. And I demonstrated for myself that that's possible. So in that summer of 2024, I decided we know very well that nutrition and exercise and weight loss results in improvements in health biomarkers. And so that's what I did. I improved my nutrition and exercise and lost weight. And accompanied with this approach was a pretty drastic improvement in my VO2 max values. So I went from below average within a month to above average. So definitely you can do it. There's an intervention and it doesn't involve anything weird. Like you don't have to take any medication. You don't have to inject yourself with stem cells to become younger. Just have to do exercise and improve your nutrition, and you're going to improve your VO2 max, aka biological age. And so, of course, what we want to do really is, can we correlate them with the underlying molecular changes? And so could we switch from this profile to another profile that's associated with these markers? And so there's many different approaches besides exercise and food. There's other types of approaches, including, as I mentioned, stem cells and other biologics, as well as, of course, pharmacological, including, for example, metformin is supposed to have anti-aging properties, and then also physical parameters like pressure, which is also associated with oxygen levels. So that's also kind of a pharmacological intervention. And so all in all, there's more than 200 of these interventions that have been proposed, which makes it super challenging to decide what should you be doing to really optimize your improvement in order to become competitive so that we can win the $100 million prize. And so at the heart of this is this question, when you combine multiple interventions, will they just keep making it better and better and better? So if I improve my VO2 max and I do this and I take metformin and I do that, will I eventually become Lamia? That's really the goal here. And so this is one of the projects that Vina is also working on, is trying to understand how can we use our molecular targetome approach to integrate structural and system level information to actually make these kind of predictions on whether more than one molecule will have synergistic or at minimum additive, but in the worst case, even antagonistic effects, right? It might be that it makes it worse if you combine two different things. And so we are running a clinical trial right now where we are combining exercise and supplements that are based on our metabolomic data together with this oxygen intervention. And so originally we formed a team called Metabolic Marvels. And there was another team in Phoenix that was an industry-led team by Clinton Hughes, the founder and CEO of this company called Youth Renewal Sciences. And he reached out and we decided instead of competing amongst the other 765 competitors, since we are both in Phoenix, we should be joining teams. And so we've changed our name from Metabolic Marvels to ASU Team Healthspan. And we are now listed amongst the top 100 teams. And about in 2 months from now, we are hoping that we can make it into the top 10 teams. But of course, we don't know. So we're working very hard on doing that right now. And this comes with really a lot of effort. So just in cost and it's just a completely crazy project where you have to be creative on a daily level. And so we have built basically infrastructure to be able to do this out of nothing. So there's no grant that supports us. So this is all creatively funded. And when we get when we get the prize, it's gonna be too late because we would have had to spend all this money already. So this is like this is a challenge in doing this kind of thing, which actually makes you creative in having to collaborate in completely new ways, involving nonprofit organizations and industry partners together with a common goal, and trying to combine these kind of commercial interests with our very fundamental scientific interests. And so with that, I know I'm 3 minutes late, sorry. I just want to thank all my collaborators, especially the recent ones that are involved in XPRIZE. Of course, my lab members with whom nothing, without whom nothing would get done, including my wonderful undergraduate students, especially from the Hours program, and 2 of whom are here today, Vina and Chrissy. Thank you, thank you. And last but not least, I want to thank the High Performance Supercomputing Center because they're absolutely amazing. I mean, ASU has the most amazing compute resources and the most amazing support. So Gil Spire is always available to help us in generating this data-driven approach. And so I can absolutely follow my desire to look at data to the fullest extent. And last but not least, here's just a picture in our facility of some of our team members in front of a 10,000-pound chamber that was delivered from Turkey and had to be disassembled and reassembled in order to make it into our facility. And if you're interested in this, feel free to contact me. Here's my email address. Thank you.
Thank you so much for that very informative and interesting talk about both your career and your research. So we learned a lot from that. And so now I will open it up. So if anyone would like to ask questions, please feel free to put them in the chat, or you can unmute and you can ask them. And we'll be having questions until around 8 p.m. Or if there's more questions, feel free to, we can go over a little bit. So while everyone's thinking, I have a question. So what do you think is the next biggest or going to be like the advancement in biological or health science? Because we have like the genome and then we have multiomics. So now what do you think is going to be next?
I mean, we are right in the midst of it, right? The next is today, and there's still so much work to be done. So we have incredible new tools with AI that we can utilize to more rapidly advance our understanding of biology and medicine. And it's really a question of how do you make use of all this data? There's just so much data, and we just have to figure out the best way to synthesize that data, and hopefully not have to do as many experiments in the lab anymore, so that we can focus in on the few that the computational approaches still cannot figure out on their own without our help.
Interesting. Data. So we have a question by Summer in the chat, and she says, if you win, what will you spend your winnings on researching next?
Yeah, that's an excellent question. And I'm a pessimist, okay? Many years of working in research, nothing ever works. And so yeah, we will, I mean, I have no doubt that, the more you learn what you don't know. And so we'll be learning so much about aging, and so we'll be spending the money on doing more research for sure.
Cool. And Chrissy under is asking, what are some tech resources beginner scientists undergraduates can use to perhaps develop new hypotheses?
Yeah, so you are welcome, and you have already experienced the hours program. So that's one approach, right? So the molecular docking that I showed you is just one of thousands of methods that people have developed for all kinds of purposes, right? So for example, I myself spend a lot of time in my career predicting protein-protein interactions and integrating databases to be able to do that. And so those methods are out there, and they're improving. And with the recent advances, there's leaps of improvements that are being made, and they generate hypotheses, and they're completely available and very easy to use. For example, I'm sure you all have heard about AlphaFold, right? And AlphaFold is the method that is very successful at predicting structures. Of course, it has limitations and all kinds of problems, but it's also useful. And all you have to do is log in with your Gmail account, and you can submit a sequence, and it spits out the structure. So it's like basically, and it's a novel structure. Nobody knew that structure before, right? You submit it, and it's a novel structure. It's like, wow, scientific discovery at the microsecond time scale. So yeah, there's a lot of tools out there that you can use, and actually the acceptance in the scientific field is shifting towards allowing purely computational combination of computational approaches to be published. Like in the early days, anytime you make a prediction, people would say, oh, that's just a prediction. And unless you have experimental data that validates that prediction, it was basically unpublishable. So I can give you a typical example for this. We predicted that rhodopsin, a protein in vision, interacts with chemokines, proteins in the immune system. At that time, it was completely unheard of. It was a protein in vision. Everybody was like, wow, really? You are weird. And so we had experimental data supporting it, but I'm not an immunologist, and I have no idea if these chemokines are even expressed in the retina, ever come there. And so even though we had hard biochemical evidence that they physically bind, I didn't know whether this would have any kind of function implication. So we never published this. And so now, decades later, there's so many papers that prove that there's so many contributions of the immune system in the functioning of the eye and dysfunction in disease. And so, sometimes it just takes time to catch up.
Cool. And Summer is asking a follow-up question. Are you focusing on more physical, aging, neurological, or both?
Physical, aging, neurological, or both. So I am a firm believer that they're all connected. And that's what XPRIZE is testing, right? So basically, we are collecting physiological markers. So we are collecting VO2 marks and others like grip strength and leg power, and so those kind of things. We're also collecting cognitive abilities through cognitive testing. And we are collecting molecular data, and especially molecular data that relates to immune function. And so basically the idea is that with the same intervention, you improve muscle, immune, and cognitive function all together. And so, so yeah, we are not focusing on one, we are focusing on the integration.
Interesting. And then next, Dahlia is asking a question. Is there an area of study that you've never had a chance to research? If not, what should we do more research in?
I won't answer that question because that you have to decide. So yeah, I mean, there's lots of interesting areas and many areas that I have not worked in. And honestly, the biggest problem I have in my research is to say no, because there are so many interesting questions that I would love to study, and the day just only has 24 hours.
Cool. And Harry's asking, are there any books or studies you've been reading recently that you would recommend?
Yeah, I'm trying to see this book. I bought this book. I can't find it right now, and I don't remember the author. Let me get back to you on that one.
Cool. And then Chrissy, I believe, raised her hand. You have another question?
Yeah, so it seems you're working a lot in the intersection of biochemistry and computational or like computer science. So if you were an undergraduate now, what would you prioritize learning?
I think that you need to follow your passion. It
Mm.
doesn't matter actually what you prioritize, it's whatever you find most fun, because research is interdisciplinary. So you will always find the boundaries of that technique and then try to interact with other people. So in this day and age, it's not possible to know all the different disciplines. You just can't, right? Like, I mean, the Renaissance times are over, where it was possible to know everything that humans had discovered to that time. Nowadays, you can't. And so, so you have to rely on domain expertise by other people and instead work with other people to collaborate with them. And you bring your approach and your viewpoint to the table, and they bring theirs, and you figure out how it best works together. And so usually, whatever angle you take, you will be able to make novel contributions. And that can be done even by yourself, right? Like, you don't even need collaborators. You can just look at any subject area from your particular view angle.
Well said, thank you.
Thank you.
I guess
And
there's also a question here on moving towards computational data. This is actually a really, really interesting question, because as I mentioned, you know, it used to be that you make predictions and the gold standard still lies with the human in the lab. And you as the human in the lab, really the traditional lab, you are the ones who has the power, because you have the resource, the methodology perfectionated in order to collect data, which is input into the computational models. The computational models can do nothing without traditional labs, because they need that data to be trained on, right? And so, and it used to be such that you had to have a lab in order to validate the predictions. Now, I feel that it is shifting to the opposite, because humans actually have a problem in that there's always errors in what we do. And so when we make mistakes, either in the experimental setup or in the interpretation of our data, you can actually use the computational approaches to potentially discover that. And I experienced an example like that, where authors of a paper had kind of hidden data in a supplement and completely twisted the conclusion from their study in a way that they wanted to see it. And it did not fit with my targetome predictions. So I was very upset and blamed my targetome predictions. And then eventually I found out that actually my targetome predictions were correct and the interpretation of the data was wrong. And so actually you can start twisting it and using the data-driven approach to cross-check the validity of lab experiments. So it's going to be a two-way street for sure.
And it's interesting too because like there's so much that it is so difficult to see and to even design an experiment to like look at a lot of these things, especially on like the molecular level. And then if you couple that with starting with a hypothesis and you've put all this, time, money, effort into, conducting these really difficult experiments, then like, yeah, you're maybe more prone to twisting your conclusions a little bit. So yeah, so it's really interesting to sort of flip the script a little bit.
Yeah, exactly. I fully agree with you.
Cool. And Hannah is asking, how do you quantify aging? For example, how would you know you have reversed aging by 20 years instead of 5, or is it more unique to an individual starting point?
So you completely hit the nail on the head. This is at the heart of this question, right? How do we quantify? And there is no agreement. Nobody agrees on what's the best way to quantify biological age. We just can't. So there's a lot of effort trying to integrate multiple different areas. So you could argue, for example, in the end of the day, I don't care what my metabolome looks like, but I do care whether I'm able, to do the upside down handstand that my daughter is able to do, right? So you can come up with age definitions based on those functional capabilities, and there's something called the intrinsic capacity that tries to do that. So try to emphasize the functional outputs rather than something that's more abstract. But in the end of the day, these functional outputs are enabled by the underlying molecular composition. And so I think it's always going to have to be a combination of these. And in all the years that I've worked on computational biology, I've only ever seen that integrating different data helps. So if you have multiple data sources, usually you can do better prediction than if you only have one. And so I think that's most likely what's going to happen here too. But in the absence of an agreed upon age definition, the best you can do is something like this. Let me show you. I have a graph here. Sorry. Where is that? Yeah, sorry, it's taking me some time. So I don't want to waste your guys' time. But basically, the answer is you can just do a simple definition by taking your one marker, whatever that is, whatever you care most about, and just plot that as a function of age. And then whatever it looks like, the assumption is that whatever the value of that marker was when you were young was the young age, and whatever the value is when you're old is your old age. And so whatever function you fit to that data is what you can use to quantify biological age. But yeah, there's no agreement as to what that marker should be. In fact, some people argue that you can use anything, any biological molecule in the end of the day can show age dependence. Just have to find the right conditions.
Very interesting. And now we're going to pivot back to the computational stuff with Summer's question. Do you see a time when all wet labs have a computational liaison to make predictions to refine experiments?
I think so. I hope so. Why wouldn't you use that resource, right? It can only make life easier.
Cool. I think that's all we have for so far. So if anyone has any other last minute questions, or if I missed your question in the chat, please put it in there. Otherwise,
I'm gonna put my email address in here. Feel free to reach out to me anytime.
yes, thank you. And also, while people are thinking for questions, if they have any, I would like to thank our advisor, Dr. Hogan, for coming today. And I would also like to thank our officers for helping support the Pre-Health Integrative Medicine Club. So thank you all for coming. And most of all, thank you, Dr. Klein, for being our guest speaker today in our first event for the spring semester. And last but not least, thank you to all the attendees today for coming this evening and being wonderful participants and supporters of the PHIM Club. So thank you all.
I would like to second that. Thank you, Dr. Hogan, and thank you, Vina, for the invitation. It was great to meet you all.
Yes, it was wonderful today. And this video will shortly be uploaded on our YouTube channel. So if anyone would like to watch this or recap, you can feel free to watch it there. And our YouTube channel is linked on our website. And also, if you would want to find more about the club, you can check out our website. And you can also become a member if you aren't already. So yeah, I think that's all I have for concluding speeches, but thank you so much, everyone. And if I don't see any other questions, so we will conclude now.
Thank you. Have a good night.